Hello everyone, myself Nisha. In this video I am going to explain about production of food yeast. Food yeast. Yeast is a single-celled living organism that transforms sugar and starch into carbon dioxide and alcohol through fermentation. Yeast is essential to baking bread and making beer and wine. Yeast scientific name is Saccharomyces cerevisiae, which quite literally means sugar-eating fungus. There are two types of yeast, baker's yeast and brewer's yeast. Baker's yeast. Baker's yeast is a type of yeast specifically used in baking to leaven bread and other baked goods. It is a single-celled organism called Saccharomyces cerevisiae. When mixed with flour, water and other ingredients, baker's yeast ferments the sugar in the dough, producing carbon dioxide gas. This gas gets trapped in the dough, causing it to rise the create a light, airy structure in the finished product. Baker's yeast secrete enzymes maltase to glucose, invertase, sucrose to glucose and fructose, zymase, sugar to carbon dioxide, ethanol. Types of baker's yeast: cream yeast, yeast slurry, compressed yeast, dehydrated yeast slurry, and cake yeast. Activate dry yeast, coarse oblong granules, live yeast cell encapsulated inside medium and dehydrate. Instant yeast, dry yeast, small granules, no need to rehydrate. Osmotolerant yeast, rabbit rice yeast, dried form of instant yeast, smaller granules and dissolve faster. Deactivate yeast, dead yeast, no leavening value. It is a powerful reducing agent, increase of dough and pizza making. Production of baker's yeast. Yeast strain selection. The process starts with selecting a suitable strains of yeast, usually Saccharomyces cerevisiae, which is commonly used in baking due to its ability to ferment sugars and produce carbon dioxide. Inoculum preparation. A small amount of yeast is inoculated into a nutrient-rich medium, typically containing sugar, nitrogen sources, vitamins, and minerals. This allows the yeast cells to multiply and grow. Fermentation. The inoculum is transferred to a larger fermentation vessel where conditions such as pH, temperature, oxygen level, and agitation are controlled to promote optimal yeast growth. During fermentation, the yeast cells consume the nutrients and multiply rapidly. Harvesting. Once the yeast culture reaches a specific cell density, it is ready for harvesting. The culture is separated from the fermentation medium through processes like centrifugation or filtration. Yeast washing. The harvested yeast cells are washed to remove any remaining fermentation medium components such as sugars and byproducts which could affect the quality of the final product. Drying. The washed yeast cells are then dried to reduce their moisture content to a level suitable for storage and use. This is often done using spray drying or fluidized bed drying methods. Brewer's yeast. Brewer's yeast is a type of yeast primarily used in brewing beer and wine. It is a single-celled fungus called Saccharomyces cerevisiae. Brewer's yeast plays a crucial role in the fermentation process, converting sugars into alcohol and carbon dioxide. Brewer's yeast is also a popular nutritional supplement due to its high protein, vitamin, and mineral content. Types of brewer's yeast. Active dry yeast. This form is dormant until rehydrated in warm water, which activates its fermentation capabilities. It's commonly used in home brewing and baking. Instant yeast, also known as quick rise yeast, it can be mixed directly with dry ingredients without the need for rehydration, making it convenient for baking. Liquid yeast. This form comes in liquid suspension and it's often used by professional brewers for its higher cell count and viability compared to dry yeast. Yeast extracts. These are concentrated forms of yeast, often used as flavorings or nutritional supplements due to their rich nutrient content. They come in various forms such as powder, flakes, or paste. Yeast cakes. These are compressed blocks of yeast cells typically used in traditional beavering methods. They require breaking apart and mixing with warm water before use. Production of beaver's yeast. Propagation. The process starts with a pure culture of yeast cells, typically Saccharomyces cerevisiae or Saccharomyces pastoriens. This culture is propagated in a sterile environment using a suitable growth medium rich in nutrients such as sugars, vitamins, and minerals. The yeast cells multiply, grow, forming a dense population. Fermentation. Once the yeast cell culture has reached a sufficient density, it is transferred to a larger fermentation vessels. Here the yeast cells continue to grow and metabolize sugars, producing alcohol and carbon dioxide as byproducts. The fermentation conditions, including temperature, pH, and oxygen levels, are carefully controlled to optimize yeast growth and productivity. Harvesting. When the fermentation process is complete, the yeast cells are separated from the fermentation broth. This can be done using various methods such as centrifugation, filtration, or sedimentation. The harvested yeast cells from a thick slurry or paste. Washing. The harvested yeast cells are washed to remove any remaining fermentation residues such as spent grains or hops. This helps improve the purity and quality of the yeast product. Drying. The washed yeast cells are then dried to reduce their moisture content and increase their shelf life. This can be done using methods such as spray drying, bed drying, or vacuum drying. The dried yeast cells are typically ground into a fine powder or compressed into pellets for storage and transportation. Thank you.